Hi, everyone. Welcome to my presentation, The Big Wonderful Immune System. I'm Heidi Gurov, and I'm the Clinical Consultant and Quality Improvement Specialist for the Wyoming Department of Health Immunization Unit. And today we are going to go on a journey through the human immune system and what it does and why it does it and how vaccines work. So I'm very excited to share this topic with you. So we're going to talk about the general anatomy and function of the human immune system, types of immunity, all the types of vaccines and how they differ, some concepts like breakthrough disease and vaccine failure, and then touch on herd immunity to wrap things up. So the study of the immune system is called immunology, and this is just the branch of biology that studies immunity in living things. So humans are not the only ones with immune systems. Our furry friends like dogs and cats have them, along with horses and polar bears and everything in between. So the immune system is not just for us. Immunology is a very complex subject. It has so many components and parts that we couldn't even begin to touch on in an hour. So I know it was probably the hardest college class I ever took. I actually cried during my final exam. It was pretty intense. So I hope over this hour that we can journey through learning about this topic together, and hopefully I can break it down for you and not cause anyone to cry while they're listening to this. So what is the immune system? It's a complex system of interacting cells that identifies foreign substances and develops a defense against these substances. This defense is known as the immune response and involves the production of specific molecules, proteins, and cells. And the immune system has two key principles. The first one is specificity, which means it is responding to a very specific thing. So every time your immune system responds to something, it's not an all-out, free for all. It's looking at like one particular protein and going, hey, we gotta do something about this. The other key principle is memory. So what this means is that once the immune system sees something, it is able to remember it in the future. So this is how we can, like if you live through natural infection with measles, you're considered immune for life because your immune system's going to remember that years down the road and have those antibodies to protect you. Some of the first things I want to define is the word antigen. You'll hear this a lot in immunology, and an antigen is a substance capable of producing an immune response. It is recognized as non-self or foreign. And there are two types. So exogenous antigens means they come from outside of the cell or the body. So this can include things like pollen. We are in the middle of allergy season and we know pollen is high and pollen is an antigen. It's something that our body recognizes as not being part of ourselves and we should do something about it. Other exogenous antigens can be things like transplanted organs and also microorganisms such as bacteria, viruses, fungi, parasites. The other type of antigen is endogenous antigens. So this means they are produced within our own cells. So this can include things like viruses. So what viruses do is they're actually not quite living, but that's a whole nother rabbit hole we could go down. But what they do is they take our own cells and they kind of hijack them and use that to reproduce and make more copies of themselves to spread out. So technically viruses can be endogenous when they're inside our own cells, but they can be exogenous if they're just like floating through the bloodstream. Other endogenous antigens can include products of normal cell metabolism, and sometimes, stuff that is really ourselves can be recognized as foreign. This is where you can get into like autoimmune conditions, stuff like that. So the immune system is made up of several different organs. So we have the tonsils and adenoids, and these are kind of found in the back of the throat area. And they help filter and remove antigens that enter through the nasal and oral routes and contain many immune cells. We have the lymphatic system and lymph nodes, and this is a network of capillaries that bring fluid back to the heart where then it rejoins circulation. And lymph nodes kind of serve as biological filter stations to help remove antigens and foster contact with the immune system. Down in our gastrointestinal tract, we have Peyer's patches and the appendix, which helps remove antigens from the GI tract. So this can be important with like antigens found in our food, whether they're pathogenic organisms or, you know, even have a play in like food allergies. Sometimes people with food allergies will have pretty severe GI symptoms. Located above the heart and behind the breastbone is the thymus. So the thymus gland is actually only fully developed in children, and from our adolescent years onward, it slowly turns into fatty tissues. In the thymus, there are cells that mature into T lymphocytes, which we'll talk about a little bit later. These are a major player in the immune system. And sometimes the thymus is called the T cell school, because it's where they kind of learn how to do their thing. Then we have the spleen. The spleen's an organ located in the upper left abdomen beneath the diaphragm. It filters and specializes in trapping bloodborne pathogens, also removes old red blood cells, stores and removes platelets, and stores defense cells. Then we have the bone marrow. This is soft tissue that is found inside of bones. And this contains stem cells for B cells, which are another big player in the immune system, T cells, red blood cells. And here, whereas the thymus was the T cell school, bone marrow is kind of the B cell school. It's where the B cells will mature. And then we have what's called mucosal associated lymphoid tissue, can be abbreviated MALT. This is the lining of the GI tract and skin, and these serve as barriers, you know, to help keep us healthy, also can contain immune cells. So if you look carefully at many of these organs of the immune system, you might begin to think they all have something in common. So one thing that many of these have in common is that a lot of these organs can be removed and are removed. So there are people who have their tonsils out. There's people who obviously their appendix goes awry. Sometimes people have to have splenectomies, whether spleen's not working or is injured in an accident. So these organs, while they are incredibly important, they're not necessarily necessary for life. Obviously, like we can't remove our heart and lungs and still live. Well, you can totally live a completely normal life without your tonsils. Many people do. Sometimes if you do have to have one of these organs removed or it's not functioning properly, you do have to take certain precautions. So like those who have a splenectomy or their spleen is not working, they have an increased incidence of bacterial infections from organisms like Neisseria meningitidis, which causes meningococcal disease, Streptococcus pneumoniae, and then Haemophilus influenzae. So things like our pneumococcal, Hib, and meningococcal vaccines are important in those without spleens because they kind of have lost that important organ that helps filter the blood. So we do have a couple different versions of immunity. The first is innate immunity. So this is just immunity that we just all you're born with it. So we have anatomical barriers. These can be both physical and chemical. So things like skin, which is the largest organ in our body, helps physically keep stuff out of the inside. We also have our mucous membranes. Then we have things like saliva and tears. Saliva and tears have an acidic pH and contain many antimicrobial proteins and peptides. We also have an innate cellular response. This can include things like phagocytic cells, like macrophages. These are kind of like the vacuum cleaners of the immune system. They just go around and gobble up stuff. We also have dendritic cells, natural killer cells. These are all immune cells that we always have. They don't need to develop a specific immunity to anything. They just are going around keeping us safe from everything. Then we have soluble proteins. So these are proteins that are part of the immune system that just help destroy antigens. So we have like the complement component pathway, cytokines, lysozyme, interferons, all sorts of different proteins. We also have a couple of other things for innate immunity, like the inflammatory response. So systemic things like fever. Fever is a very important part of the immune response. I know sometimes we're all quick to jump on and try to control fevers, but a lot of times a low-grade fever is beneficial because it's your body's natural response to an antigen. We also have things like the cough reflex and sneezing. Both of those things are methods to help remove antigens, pathogens from the respiratory tract. That's why I am huge on, if you got to sneeze, you can't hold it in. You got to let it out because that's stuff your body is trying to get rid of. It's kind of an inside joke in the office because I always hassle one of my coworkers about her stifling sneezes. I'm like, no, let it out. So she'll get a kick out of that. I included that. At least I'm not naming names. So the other type of immunity is called adaptive immunity. So adaptive immunity has a really high degree of specificity with memory. So adaptive immunity is responding to a very specific antigen, and then it's remembering that for in the future. Adaptive immunity is going to have self, non-self recognition. So it's going to know what is normal in our own body and then what is foreign. Adaptive immunity is not independent from innate immunity. In fact, it is pretty poorly effective without innate. So innate and adaptive immunity are not completely separate. Obviously, if you're having, let's say you have measles, you're having that adaptive immunity, but then you also might be having some of that innate stuff like a fever going on. Adaptive immunity has two types, humoral versus cell-mediated, which we will talk about, and then it can be active versus passive, which we will discuss later as well. So we'll start with humoral immunity. So this is named because it involves substances found in the humors or body fluids. It's kind of a historical term where before modern medicine, people thought disease was caused by an imbalance in the humors, like you had different body fluids that meant different things and you had to balance them out. So some historical background on why it is called such. So with humoral immunity, our big players here are the B lymphocytes or the B cells. These cells mature in the bone marrow. And they do have a couple different types. We have memory B cells, which have a very long lifespan and have things called antibodies that are bound on their membranes. Then we have plasma cells, also known as effector B cells. And these cells only live a few days, but what they do is they actually secrete the antibodies. And then we have 5 different types of antibodies. Humoral immunity is especially important against bacteria, parasites, toxins, and viruses that are outside of the cells. So like those that are traveling through the bloodstream and are not hijacking the cells to reproduce. With humoral immunity, antibodies bind to antigens circulating in the lymph or blood and then neutralize it and facilitate its elimination. So a little bit about how this all works. So first, an antigen is going to bind to a B cell. Like that little B cell is going to like, hey, this fits with me. And that antigen's like, ooh, I fit with this. Although the antigen probably doesn't realize it means it's going to get killed. So the B cell and antigen is going to kind of float around. And then a helper T cell is going to come by and help activate it. So we're going to talk about T cells in a little bit. It's kind of hard to split these into two things, so bear with me. But the T cell is going to secrete different molecules and chemicals that's going to help excite that B cell. And then after it's all activated and excited, that B cell is going to divide, and it's going to produce both plasma cells and memory cells. So those B cells that are plasma cells are going to produce antibodies that are going to attach to the current type of antigen or invader. And these antibodies are going to help facilitate it getting killed off and cleaned up by the immune system. So like eater cells, which are like the macrophages or the vacuum cleaners, are going to come along. They're going to see these antigen particles that were marked with antibodies and eat them all up, gobble them up. The memory cells that are formed are going to float around there with their antibodies, and then in the future, if that antigen is contacted again, those memory cells they're going to mate up, and those memory cells are going to remember it, and then it'll already have those antibodies in place, and it'll help the immune system activate much faster. So what are these antibodies? So antibodies can also be called immunoglobulins, and they are a Y-shaped protein with a site for the antigen to bind to, and they really just help mark them and facilitate their destruction. There are 5 types of antibodies. So the first type is IgG. This is the most abundant type and forms the majority of antibody-based immunity. IgG antibodies can also cross the placenta from mother to fetus, which helps give that newborn baby some tiny bit of protection when they're first born. The next type is IgM. This is the first type of antibody produced in the primary response. So the first time the immune system comes in contact with that antigen, this is the antibody type that's going to be produced. The next type is IgA. IgA antibodies are found on mucous membranes, so like those in the GI tract, respiratory tract, and urogenital tracts. They're also found in saliva, tears, and breast milk. They help form an important line of defense against certain pathogens, especially those that may be like foodborne. IgE antibodies really have their purpose in hypersensitivity reactions or allergic reactions. They help trigger histamine release. They're also very important in helping fight parasitic worm infections. So anyone with allergies, you can blame their IgE antibodies. And then our final type of antibody is IgD. So this is an antigen receptor on mature B cells, and actually is not well understood what these antibodies do. So the field of immunology is actually quite young. We're looking at 1960s, 1970s when it really started to become its own branch of biology. So we're still figuring out a lot of stuff with the immune system, and IgD remains one of our mysteries. But it is believed they help activate basophils and mast cells to produce antimicrobial factors. So these are just different types of cells in the immune system. Hopefully, maybe one day we'll solve this mystery and we'll know a little bit more about IgD. So with humoral immunity and the antibody response, we have 2 phases of response, the primary versus secondary. So the primary response is with the initial encounter with that antigen. So let's say, we will use a timely example here. So COVID-19, you are exposed to COVID-19. So that initial response to that, this is brand new thing your immune system has never seen. And it's going to take a few days for the immune system to respond and make antibodies to that specific antigen. Usually takes about 5 to 7 days. So then you're going to have those antibodies in your body. And then the second time you are exposed, or future times, there would be this secondary immune response. With secondary immune response, because there's those memory B cells, there's usually a much more swift immune response with a lot of specificity. Usually it'll be like 1 to 2 days for the immune system starts responding. And there'll be a lot higher antibody levels, and this response is sustained for much longer. So that's why sometimes you can fight off infections a lot faster the second time, because you have that memory in your body. So now that we've talked about humoral immunity, we're going to talk about cell-mediated immunity. Like I said, these two types of immunity are not separate from each other. They both interact, but you kind of have to present them separately, but you're going to be hearing about B cells. So with cell-mediated immunity, our major players are T lymphocytes. So these T cells arise in the bone marrow, but then travel and mature in the thymus, or that T cell school. There are two types of T cells. We have T helper CD4 cells. These are one of the things you might have heard the term CD4 before with like HIV infections, because they do CD4 cell counts. So that's what they're trying to count with that, these T helper cells. So T helper cells recognize and interact with antigen molecule complexes. And once they become activated, they secrete chemicals called cytokines. The other type of T cell is T cytotoxic CD8 cells. So these exhibit cell killing activity once activated. I always think that's a great term, like cell killing activity. So with cell-mediated immunity, this response does not involve antibodies, but rather the activation of various immune cells in response to the antigen. Where cell-mediated immunity is important is virus-infected cells. So these are cells where the virus has hijacked them and is using the machinery of the cell to reproduce more copies of itself. Also cancer cells, and then transplanted cells, like if someone has an organ transplant, there are really a lot of those immune suppression, like anti-rejection drugs, are really trying to fight the cell-mediated immunity response. So the cell-mediated response is quite complicated. I have trouble wrapping my head around it. But so the main principle is it binds only antigens which are present on cell membrane proteins called major histocompatibility complexes. So there are two different classes of these. So we have class 1, and these are found on those CD8 cytotoxic, those killer T cells. And when it comes across one of these, it will go ahead and kill it. The class 2 are present on the surface of antigen presenting cells like T helper cells. And when it comes across this, different cytokines and interleukins, chemicals and proteins are secreted. And this is what stimulates those B cells. This is what, if you recall back to a couple slides ago, those B cells become activated and all excited. And that's because this T helper cell came along and helped them by secreting interleukins and cytokines, and the B cell will now produce antibodies, and this will bring in those macrophages, or those vacuum cleaner cells, to gobble them up. I've mentioned the term antigen presenting cells a couple times, so this can be things like the B cells, also macrophages and dendritic cells. These are just different cells found in the immune system. So yeah, kind of a complex thing. There is no need to really know the nitty-gritty about this, but pretty much you have two different classes. One class is going to be activating those cytotoxic T cells, or those killer T cells, and the other class is going to activate those T helper cells, which is going to go help those B cells that make antibodies. Big picture of that. Hopefully I didn't make that too confusing. I feel confused myself now. But. So we had the humoral versus cell mediated immunity, but now I want to talk about passive versus active immunity. So these are the two basic mechanisms for acquiring immunity. The first one, passive immunity, is protection by products produced by an animal or human that are transferred to another human. Often when you have immunity in this method, protection wanes, which means it wears off and goes away, often within a few weeks or months. So one of the most common forms of passive immunity is maternal antibodies. So these are antibodies that are transported across the placenta during the last 1 to 2 months of pregnancy. A full-term infant will usually have the same protection as its mother and may be protected from certain diseases up to 1 year. Protection is better against some diseases than others. So things like measles and rubella, they might have a better protection than from things like pertussis. Another way to get active immunity is from blood products. So many blood products contain antibodies. So whether it's a whole blood transfusion, plasma, platelets, or immunoglobulin. So you can also receive pooled antibody. And this is where it's antibodies to either many different things or a very specific disease like hepatitis B, tetanus, rabies, or varicella. So pooled antibodies sometimes are used for people who are severely immunocompromised, like the people that really don't have a functioning immune system. They will get routine injections of pooled antibody that's obtained from plasma donors, and that helps give them a bit of protection from everything out there. The very specific ones like rabies is used in like post-exposure. Let's say you were bitten by an animal that might be rabid. We'll use that pooled antibody for rabies to help really boost your immune system and that immune response. We also have things like antitoxins. Antitoxins are produced in animals, usually horses, and they contain antibodies against only one antigen. So some common antitoxins that we use include those for tetanus, diphtheria, and also botulism. The thing with antitoxins is you can have something called serum sickness result, which is an immune reaction to any horse proteins. Horse proteins are obviously going to be recognized as foreign to a human, so this is a possible side effect. We also have other medications. It seems like there's more and more of these coming out every day. Monoclonal antibodies. These are medications that often end in MAB, so M-A-B. One I can think of, like off the top of my head, is one called eculizumab. It helps treat some very rare disorders that can, and taking this medication puts you at higher risk for like meningococcal disease, but that's a MAB, a monoclonal antibody. These are usually used in the diagnosis and therapy of certain cancers and autoimmune and infectious diseases. So then we have active immunity, and active immunity is protection produced by a human's own immune system. So it's your own immunity that you have made yourself. This immunity lasts for many years, sometimes a lifetime. So much better than passive immunity. I mean, this is something that you are going to have as long as you live for certain things. So you can get active immunity in 2 ways. One is to survive infection with the disease-causing form of the organism. So in general, once a person recovers from an infectious disease, they will have lifelong immunity to that disease. This isn't every single disease out there. You have things like tetanus. If you survive tetanus, you are not immune to tetanus and still should be vaccinated. But for most things, like when you're looking at like varicella, measles, diseases like that, usually hepatitis B, you might have lifelong immunity. You're going to have really good memory. Memory B cells will circulate in the blood and also reside in the bone marrow. And upon re-exposure to that antigen, the memory cells are going to replicate and produce antibodies really rapidly to help reestablish protection. Of course, we know having the actual disease comes with a lot of risks. You might have disability, scarring, other complications, and the possibility that you might die from it. So the other way that you can gain active immunity is through vaccination. So this is the administration of antigenic material to stimulate a person's immune system to develop adaptive immunity to that pathogen. This often produces an immune response similar to that produced by natural infection, but you're not subjected to all those complications and possibly death from that disease. Often, sometimes too, you might have the memory similar to having natural disease. So there are some vaccinations that you receive that they've shown that, 20, 30, 40 years, you still have those antibodies protecting you. So normally if this was live, I'd be stopping here to take questions, but since this is recorded, if you do have any questions or comments or anything, definitely shoot me an email or you can give me a phone call and I will help you out. Like I've said in all my presentations, I wanted to keep all my question slides because I think sometimes I find clever photos. So here's one from the Awkward Yeti, a funny little one about macrophages and antigens. So next up, we're going to talk about vaccines. So vaccination is a form of active immunity, as we just discussed. So sometimes you kind of hear like the words vaccination versus immunization used kind of interchangeably. There's actually different definitions for both. So immunization is the act of making a person or animal's immune to infection, typically by vaccination. So vaccination is the act of taking the vaccine and injecting it or however you administer that vaccine. And then immunization is actually when the person forms an immune response. Kind of funny. I know I'm guilty even using vaccination, immunization interchangeable, but they do have two different definitions. Usually with vaccination, immunity and immunologic memory is similar to natural infection, but once again, without the risks that come along with the actual disease. And we have two types of vaccines. We have inactivated and then live attenuated. And then as a general rule, the more similar a vaccine is to the disease-causing form of the pathogen, the better the immune response is to the vaccine. There are different factors that will influence the immune response to a vaccine, things like the presence of maternal antibodies. So we don't vaccinate for MMR varicella until 12 months of age. And this is because infants may have that maternal antibody still circulating, and that antibody will interfere with those live vaccines. Also, the route of administration makes a difference, and the presence of an adjuvant, which is something that is added to the vaccine to increase the immune response. We'll discuss them a little bit later. Also, things such as host factors, such as age. We know, like sometimes in the very young or the very old, they do not respond to a vaccine as well as maybe a 25-year-old. Also, nutritional factors, genetics, and coexisting diseases can all play a role. So first, we'll talk about inactivated vaccines. Sometimes you might hear them called killed vaccines. So in an inactivated vaccine, there's nothing living. It cannot cause any form of disease. This includes flu shots. There's nothing living in a flu shot that can give you the flu. So inactivated vaccines are made by growing the organism in culture media and then usually inactivating it with heat or chemicals. Sometimes the organism is further treated to purify it down to only certain components, so it doesn't have the whole like virus or bacteria in it anymore. These are less affected by circulating antibodies, which is why the vaccines given, to young babies are usually inactivated vaccines, because those maternal antibodies are not going to impact it. Inactivated vaccines always require multiple doses. In general, the first dose primes the immune system. It kind of goes, hey, immune system, look at me. And the immune system starts looking at it a little bit like, what are you? And then the protective immune response develops after the second, third, or any further doses. So an example of this, we can look at like acellular pertussis vaccine. So you have about 68% efficacy after the first dose. This means how well it protects. After the second dose, this jumps to about 92% and over 99% after that third dose. Like I said, that first dose just kind of wakes up the immune system, and then that really strong antibody response happens in those future doses. Response to inactivated vaccines are a humoral response. So you're looking at antibodies with those B cells, also those T helper cells helping excite those B cells. You'll get little to no cellular immunity response, and antibody titers will diminish with time. So this is why some inactivated vaccines we do booster doses on. A great example of this is TD or Tdap, that every 10 years you should go get another booster to help keep the immunity up for tetanus, diphtheria, and pertussis. So there are several different types of inactivated vaccines. So we have whole cell. So whole cell is exactly what it's called. It contains the entire whole cell that's been killed. Vaccines that are available in the US that are whole cell include polio, hepatitis A, and rabies. There's been a lot of other ones in the past that are no longer available. An example of this would be the whole cell pertussis that was used throughout like the 1980s, early 1990s in the US. We also have fractional. So what fractional vaccines are, they just contain pieces of the bacteria or virus that are antigenic and stimulate the immune system. So we have toxoid. These are based on toxins that the bacteria produce that have been inactivated. And we usually need several doses of these vaccines and an adjuvant to really help our immune systems respond to them. So an example of this is diphtheria and tetanus. We have the subunit vaccines, which only contain antigenic parts of the pathogens. They've kind of taken out all that excess stuff that doesn't stimulate the immune system. We see this with hepatitis B, influenza, acellular pertussis, and anthrax. Other types of fractional inactivated vaccines include the pure polysaccharide based. These are a unique type of vaccine that are composed of long chains of sugar molecules that make up the surface capsule of the bacteria. So certain bacteria kind of have this sticky, sugary coating around them that helps them evade the immune system. And these pure polysaccharide vaccines take advantage of that. Examples of pure polysaccharide vaccines include typhoid, the injectable version, and then our pneumococcal. So PPSV 23 or Pneumovax 23 is the polysaccharide version of the pneumococcal vaccine. The thing with the pure polysaccharide vaccines is that the immune response is usually independent of T cells, which means these vaccines are able to stimulate B cells without the assistance of T helper cells. This is kind of an interesting, unique thing. But the problem with this is they do not consistently work in those younger than 2 years of age, because when you're younger than 2, you have a very immature immune system. If you give repeated doses of these pure polysaccharides, you actually don't get a booster response. Most of the time when you boost a vaccine, you get huge antibody titers, and this doesn't happen with these vaccines. The antibody induced usually has less functional activity than that induced by protein antigens. And the antibody response is usually IgM, which are very short-lasting antibodies versus the IgG that can last a very long time. So we don't see these vaccines used at all under the age of 2 for that reason. So a much better version of this is the conjugate polysaccharide vaccines. So in the 1980s, they kind of realized, these pure polysaccharides had some problems, especially in young children. So they made conjugate vaccines, which means the polysaccharide is chemically combined with a protein molecule, which is often diphtheria or tetanus toxoid. This changes the immune response to a T cell dependent, and it means these vaccines work well in infants and you get a booster response to multiple doses. So really good antibody response. So our conjugate polysaccharide vaccines that we use include Hib, pneumococcal, PCV13, Prevnar 13, and the meningococcal vaccines, Menactor and Menbeo. Now our final type of fractional vaccine is recombinant. So recombinant vaccines are produced by genetic engineering technology. So as research techniques have gotten better and vaccinology has, had decades and decades of research and study behind it, a lot of vaccine technology has gotten really high tech. So these recombinant vaccines use modern genetic engineering technology. And sometimes they'll insert a segment of the virus's DNA into the gene of a yeast cell. An example, recombinant vaccines include hepatitis B, HPV, and influenza. So a little less complicated are the live attenuated vaccines. So these are derived from wild viruses or bacteria that are weakened in the laboratory. Usually they are grown through a repeated cell culture technique. So the measles vaccine that we use, the virus is from a child in the from 1954, and they've just used that. They've been able just to keep it going in cell culture. It's kind of cool. So the thing about live attenuated vaccines is they are very fragile because they are technically living. So anything that damages the live organism, such as heat or light, or interferes with the replication of the organism in the body, can cause the vaccine to be ineffective. So this is why, which for all vaccines, storage and handling, proper storage and handling is very important. They're especially important with our live attenuated vaccines. Because if you kill the living virus that's in that vaccine is going to be ineffective when it is given to a person. So the immune response to live attenuated vaccines is virtually identical to that produced by natural infections. So you get the antibodies, the T cell response. Usually immunity will be produced with one dose. You get a very long prolonged immune system exposure. So you get increased production of memory cells. The thing with this though is that there can be interference from circulating antibody. So this can include those maternal antibodies that infants have. Also transfusions. So if you get a blood transfusion, that's going to have all the antibodies that the donor had. So that can interfere with any vaccines. Also other live vaccines. That's why we have minimal interval rules between two different live vaccines. Live attenuated vaccines also cannot be used in immunocompromised persons. There's a possibility of severe reactions because there might be uncontrolled growth of the vaccine virus. So those, with severe immunodeficiencies cannot receive live vaccines. Also theoretically, a live attenuated vaccine strain can revert to the pathogenic form. This is only known to happen with live oral polio vaccine, which is kind of a driving reason of why we stopped using oral polio vaccine in the United States, because we didn't want any vaccine-derived strains out there possibly reverting back to wild-type polio. So our live attenuated vaccines include measles, mumps, and rubella, known as MMR, also varicella. There is a live version of zoster. There's rotavirus, intranasal influenza, yellow fever, oral typhoid, and vaccinia, or also known as smallpox vaccine. So we know there's lots of different parts that go into making a vaccine. You just don't inject an antigen and call it good. There's all sorts of different ingredients. And sometimes these components and ingredients can be the cause of a little bit of controversy and possibly a lot of questions from patients and parents. So the main ingredient, of course, is the antigen. This is the thing that stimulates the immune response. It can either be that whole cell or those pieces that have been taken out through higher laboratory techniques. We also see stabilizers. So stabilizers help maintain effectiveness and storage. We know a vaccine isn't produced and then immediately given to someone. You know, they are shipped to your clinic and stored in the freezer and fridge. So you gotta have something to keep it effective. Some of these include magnesium chloride, magnesium sulfate, and lactose sorbitol. Another stabilizer that may be used is gelatin. So we know gelatin is an animal product. I do like to say all religious groups have approved the use of gelatin containing vaccines for their followers. So in case that question comes up, they've all approved it. Another group that may have some objections to gelatin are vegans because they do abstain from any animal products. Another vaccine component is antibiotics. So antibiotics are usually there to help prevent bacterial contamination of tissue cultures that are used during the manufacturing process. Common antibiotics found in vaccines include neomycin, streptomycin, polymyxin B, chlorotetracycline, and amphotericin B. Luckily, these antibiotics rarely cause human allergies, definitely not like a penicillin allergy that can be fairly common. These are very, very rare allergies. So it might be very uncommon to run across a person that has like a known neomycin allergy. Our next component is adjuvants. So these are things that help stimulate the production of antibodies against the antigen. These make vaccines more effective by enhancing, accelerating, and prolonging the immune response. And adjuvants are incredibly important in inactivated vaccines. And there are several hundred different types. There's new ones being developed all the time, which are like new novel adjuvants. I know like Shingrix, the newer inactivated shingles vaccine that came out recently, uses like one of the newer adjuvants, which is why it causes such an amazing immune response in people. So one adjuvant that gets kind of a lot of attention is aluminum. So aluminum has been used as an adjuvant in vaccines since the 1930s. So it's one of our oldest adjuvants. It's very proven and studied. So aluminum is a natural element that is present in our environment. It is found in our food and water and air, and it's something that we naturally deal with every moment of our lives. The quantity of aluminum found in vaccines is incredibly small. It doesn't take much to have its purpose and stimulating the immune system. So in comparison, in the first 6 months of life, babies receive about 4 milligrams of aluminum if they get all the recommended doses of vaccines. If they're breastfed, they will ingest 10 milligrams. If they're fed regular formula, 40 milligrams. And if they're fed like a dairy-free soy-based formula, they'll ingest 120 milligrams. Aluminum is processed the same in the body, whether it's ingested or injected. So I know aluminum is one of the common controversial ingredients in vaccines, but this really goes to show the amount is very small, especially if they are being formula fed, they will encounter a lot more of it. So the aluminum in vaccines is not unsafe. It's been very well studied, and aluminum definitely isn't an unnatural chemical for humans to come in contact with. Another vaccine component are preservatives. So these are usually added to multi-dose vials to help prevent bacterial or fungal growth, because we know you are accessing that multi-dose vial repeatedly, usually for like 10 doses. One of these preservatives that used to be used was called thimerosal. So this is an ethyl mercury compound, and it's a naturally occurring element found in the environment. It was pulled from all routine childhood vaccines in the late 1990s over concerns that were voiced between a connection between mercury and autism, which of course we know there's been countless studies that proves there is no connection between mercury in vaccines and autism, but they proactively just pulled the thimerosal from all childhood vaccines. So mercury, most people know it can be bad, like if you eat too much tuna fish or something. So what we're concerned about with that is actually methyl mercury. So methyl mercury is a form of mercury that you will find like in seafood or fish products, and this will actually accumulate in the body and cause problems. Thimerosal was an ethyl mercury compound, so this form of mercury actually does not accumulate in the body. This is kind of like the difference between rubbing alcohol and, let's say, the alcohol found in a glass of wine. Two different versions, but one is definitely not safe to consume. That's kind of like the comparison with ethyl and methyl mercury. But luckily, not something we really have to be concerned about because it is not used in childhood vaccines anymore. So that's definitely good information to share in case someone has concerns about mercury in vaccines. Another preservative that is used is formaldehyde. Formaldehyde is usually used to inactivate viruses and detoxify bacterial toxins. Usually during a purification process that occurs during production, almost all the formaldehyde in vaccines is removed. Formaldehyde is a byproduct of protein and DNA synthesis in our own bodies, so it is commonly found in the bloodstream normally, like as we're sitting here, we got formaldehyde in our bloodstreams, and the quantity found in the blood is 10 times greater than that found in any vaccine. So once again, it's kind of debunking some of those myths that these are unnatural chemicals that have no place in or on humans, and really they are things we deal with every day. Our final component I want to talk about is fetal cells. So viruses require cells to grow and to replicate and make more copies of themselves. So cell lines are used to make, fetal cell lines are used to make rubella, varicella, hepatitis A, shingles, and rabies vaccines. Human cells, obviously if it's a human virus, they grow best in human cells, better than animal cell lines that are used. So back in the early 1960s, cells were obtained from 2 elective abortions that were performed, and these cells are used to this day in cell culture for making these vaccines listed above. These cell lines do not require ongoing abortions. The same cells have been used since the 1960s. Cell lines can be immortal and just replicate indefinitely. So definitely it's not like you have to have repeated abortions to keep them going. Of course, this can be a concern for certain people who, of certain religions or moral beliefs about abortions. I would like to say the National Catholic Bioethics Center has determined the use of vaccines grown in fetal cells from historic abortions are morally acceptable to receive, usually based on some principles of the good of the greater whole. So once again, if someone has concerns about this, you can point them in the right direction on some of those stances that have been taken by their religion, and kind of explain that it's not an ongoing abortion process going on to make these vaccines. Another quick question slide before we dive into the last little bit of this presentation. So I briefly wanted to touch on breakthrough disease. So breakthrough disease is disease that happens even though a person has been vaccinated. Luckily, usually it's significantly milder than natural disease, and it can result from several factors. So with pertussis, people can either be asymptomatic or have a very mild cough illness. And this is usually due through waning immunity. Our acellular pertussis vaccines are not exactly the most efficacious vaccines. They're not as good as whole cell pertussis. So immunity can wane fairly quickly. And this is where you can see breakthrough pertussis. Unfortunately, people can transmit the disease to susceptible persons, which is why we encourage that like cocooning around infants, making sure everyone has Tdap vaccines so they have the most high antibody levels and they're not inadvertently spreading pertussis asymptomatically to that little baby. Another disease we see this for is polio. This is followed a live oral polio vaccine. Once again, we don't use OPV anymore in the United States, but it is fairly common, especially in the developing world, because it's a lot easier to use and administer than injected vaccine. So what happens here is a mutation of the vaccine virus occurs to a more pathogenic form and can possibly cause paralytic disease. It is very, very rare for it to happen, but it can occur. I decided to give varicella its own slide because I found this great infographic. So breakthrough varicella is an infection that occurs in a varicella vaccinated person, usually 42 days after vaccination. It can occur in up to 15 to 20% of vaccinated children. With breakthrough varicella, they will have very few lesions, usually less than 50, and remain afebrile and have a very short duration of illness. Usually breakthrough disease can result from several different factors. This can include interference of virus replication from circulating antibodies. So like the vaccine was given too young, given younger than 12 months of age, or they received an MMR, a week earlier and then varicella. We know live vaccines have to be spaced 28 days apart unless given on the same day. It can also result from less effective vaccine resulting from storage and handling errors. This is why we are so big on proper storage units, setting them up correctly, using data loggers, monitoring temperatures, because we have to make sure these vaccines are actually kept viable. It can also be due to inaccurate record keeping, from, once again, not knowing when previous doses were received. Usually a second dose will reduce the risk of breakthrough. So after you receive that second dose of varicella at kindergarten age, it's very rare to have breakthrough disease. Next, we'll talk about vaccine failure. We know not everything in this world is 100%, and of course this applies to vaccines as well. So usually with vaccine failure, it's really the circumstances that fail and not the vaccine itself. Like I said, there's all sorts of factors that can come into play with this. So with primary failure, this is when the body and the immune system fails to launch an adequate immune response. This could result from interference of virus replication, once again, like maternal antibodies, recent blood transfusion or blood products, less effective vaccine resulting from storage and handling errors, inaccurate record keeping. Usually with primary failure, the person will respond to a second dose of the vaccine and any future doses. A vaccine we sometimes hear about this happening with is hepatitis B. So like after 3 doses, someone will get titers and won't show immunity. In that case, it is recommended to give a second 3-dose series and then test them again. If they're still not responding to the vaccine, it is actually recommended to check them for a chronic hepatitis B infection. So if you have hepatitis B and you have it like chronically, your vaccination series is not going to do anything for you. So this is a case where you need to actually be looking for hidden disease that they might not know about. Hepatitis B is incredibly endemic, especially in Asia, and a good chunk of the population actually is chronically infected and might not even know it. So something to consider if you're seeing a failure to mount an immune response to hepatitis B after several completions of the series. Sometimes people will think they have a vaccine failure because they receive titers to something and there's, it comes back. This isn't always the case. Sometimes we refer to titering as looking for a problem that does not exist. Many times the people will still have antibody, and it's not sensitive enough to be detected by these commercial tests, but if they were to come in contact with that disease, the memory is there and they will have a protective immune response. A big one for this is varicella. Most varicella titer tests that are commercially available are not sensitive enough to detect immunity that is due to varicella vaccination. And now that we have a good chunk of our population that has their varicella immunity thanks to vaccination, which started about the mid-90s, this could be a problem that results. If you do have any further questions on that, like how to interpret titers and when titering is appropriate, definitely reach out to me and I can provide you with the CDC recommendations for titering, especially in healthcare workers, and when it is recommended and when it is not recommended. So then there's secondary vaccine failure, and what this is usually just the gradual loss of immunity after the initial immune response over a period of time. This is called waning. So this often occurs years down the road, and this is why we give boosters of inactivated vaccines. So like TD or Tdap, over time your immunity to those diseases wane, and that's why we give a booster dose of vaccine. So our last big concept that we're going to end on is herd immunity. This is also called community immunity. So what this is if the majority of the population is immune to an infectious agent, the chance of a susceptible individual contacting an infected individual is so low that the susceptible one is not likely to become infected. So herd immunity is very, very important because it helps protect those in a community that are unable to be vaccinated. So like babies, we know they, aside from hepatitis B, they cannot start vaccines until 2 months of age, and then some vaccines even later, like MMR starts at 12 months. There's also people with weak immune systems that cannot receive these vaccines. And this could be due to disease like cancer or medical treatments or chemo, or just having really severe immunodeficiencies. Also, pregnant women can't be vaccinated for everything, and we know pregnancy is an immunocompromising condition. Also, senior citizens whose immune systems just aren't as working as well. And then anyone who, let's say, is has a contraindication to a vaccine, like they have an anaphylactic allergy to a vaccine ingredient. So this herd immunity helps protect those people that are unable to get vaccinated themselves. So herd immunity, there's different thresholds that have to be met for each disease. So for diseases like measles, which is probably one of the most infectious diseases that is out there, and then pertussis, you need about 90% to 95% of people in your community to be vaccinated to shield the people most at risk. These are very high numbers that many, many places are not meeting, which is why we have seen pockets of outbreaks of both measles and pertussis. For some other diseases like polio, the herd immunity threshold is lower, like 80 to 85% might be enough to sustain herd immunity. Herd immunity thresholds really can depend on transmission and how contagious that organism is. An example of a breakdown that we saw with herd immunity was in the early 1990s when the Soviet Union was splitting up. Of course, we know all sorts of things were in jeopardy and limbo during that time as we saw all these new countries, break off onto their own, and a lot of healthcare infrastructure and immunization programs ceased to exist anymore. So there was this massive outbreak of diphtheria. So there was really only like 800 cases of diphtheria in 1989. But then in 1994, so after all this changes occurred in this area of the world, there was almost 50,000 cases of diphtheria and like 1,800 deaths. So a big factor in this was that immunization programs and mass vaccination campaigns ceased to exist, and they saw a huge rise in diphtheria. A more modern day example of this that we might remember, 2019 measles outbreak here in the United States. We saw communities with very, very low immunization rates be very affected by measles cases that were imported from international travel, and it spread through the community. This is a current worry as we live through this COVID-19 pandemic, that we are going to see breakdowns in herd immunity as well, because we know many clinics have competing priorities, or parents aren't wanting to bring their children in and possibly expose them to COVID to receive their immunizations. And we are seeing a decrease in vaccine ordering and vaccine administration, and coverage levels decreasing. So that is why the CDC and AAP and other people are encouraging, especially children under the age of 2, to try to keep them up to date on their immunizations during this pandemic. Because if herd immunity starts breaking down, you know, it's quite possible we could see like measles and pertussis outbreaks on top of the COVID-19 situation. So I have a very relevant example of why we need to do what we can to maintain our immunization programs as we go through these difficult times. So I just want to conclude with the statement that the immune system is awesome. The immune system is incredibly powerful. It does so many amazing things that it's kind of hard to wrap your brain around. So about 100 trillion bacteria live in and on our bodies at every moment. We have more bacteria on our bodies than our own cells. We have about 35 trillion of our own cells, and we encounter countless antigenic threats every single moment of our lives. Like, I'm sitting here with my kitchen window open right now, and I know all this pollen is just coming in and attacking my body. And I do know the immune system right now, it's responding to that pollen. The immune system can respond to over 10,000 different antigens. It's crazy what it can do. About 2 billion T helper cells are replenished every single day in our bodies. And really, vaccines are a drop in the ocean, but they are a mighty drop. They help aid our immune systems in creating that memory and immunity that are going to protect us for a very long time. So it's awesome, it's powerful, it can respond to so many things at a different time. And just want to say, who else loves their immune system? Antibody? Antibody? Always gotta love a good nerdy science pun. So I just want to say thank you for listening to this presentation. I hope you took something from it, and I hope I was able to break down these concepts a little bit. Like I said, it is definitely hard and complex topic, just because the immune system does so much. If you have any questions or comments or anything, definitely give me a call or send me an email. I highly encourage you to check out all the other recorded presentations for our virtual conference. And I know I'm super sad I can't see everyone in person, but I hope in 2021 we'll be able to meet again and do an in-person conference. And in the meantime, stay safe and stay healthy. Bye.